GetGenome is a non-profit organization, and really central to GetGenome is empowerment of researchers and also celebrating the talent that is around the world and celebrating collaboration and promoting that. Today, I'll just describe a small snapshot of the work that we do, and the focus will be on our work in Tunisia. But feel free to explore the website and you'll find that we've done a lot of work around the world. Just a brief introduction. I obtained my PhD in molecular biology and genetics at the John Innes Centre, which is our sister institute here on the Norwich Research Park. And I then started a role as entrepreneur in residence at the Sainsbury Laboratory, where I operate and run the day-to-day of GetGenome. So I'm joined by a number of collaborators, Tunisian researchers, and I'd like to introduce them for you.
My name is Rakia Chouari. I'm a Tunisian scientist. I have a PhD in microbiology, and I'm an associate professor at the Faculty of Sciences of Bizerte. So I'm teaching microbiology, and I'm also a member of the laboratory of plant toxicology and environmental microbiology.
Okay, so hi everyone, I am Sawsan Ben Kader. I'm an assistant professor in the High Agronomic Institute of Shat Mariam, which also located in Sousse.
Dr. Fatma Dries. I'm assistant professor at the Center of Biotechnology of Sfax in Tunisia. I have my PhD degree in biology engineering. I'm principal investigator of a national project.
I'm Manisha Rishi. I'm a postdoctoral researcher at the Laboratory of Resilience and Sustainable Agro-System, as well as the Laboratory of Bioactive Substances in the Center of Biotechnology in Sevilla in Tunisia.
My name is Emeni Blalaya. I'm a PhD student at the Laboratory of Bioactive Substances in the Center of Biotechnology in Boursa, and I'm affiliated to the University of Science in Tunis Al Manar.
In preparation for this talk, it really gave me an opportunity to reflect on the amazing interactions that I've had with a whole plethora of really amazing, dynamic Tunisian researchers over the 2 to 3 years that we've been working in the country. So it's not just the 5 that are joining us today, but dozens and if not hundreds of Tunisian researchers that we've had some interaction with. So GetGenome is genomics for all, but really it started with just one. Professor Sofiane Kamoun, who is an eminent Tunisian researcher, as a group leader here at the Sainsbury Laboratory, and is GetGenome trustee and founder. Sofiane was sitting on an examination board of Manel, a PhD student at the time, who was trying to identify and isolate and amplify a small section of DNA within her organism of interest. And she was having difficulty doing that. And Sofiane realized at that moment that the application of some state-of-the-art technology, an investment of just a few dozen pounds, and he could sequence the whole genome of that organism and provide it to Manel. And as it transpired, that piece of DNA that Manel was trying to isolate didn't actually exist in the organism. And that led Sofiane to think, well, how many more early career scientists held back by not having access to these technologies or the associated training? So GetGenome is based at the Norwich Research Park, and we are one of a number of institutes on the park. The Sainsbury Laboratory is working toward plant health discovery and plant and microbial genomics. We have a long history of genetics and genomics here in Norwich and within our institutes. Professor William Bateson was the first director of the John Innes Institute, and in a letter in 1908 coined the term genetics. But William Bateson would never have imagined the wonder and complexity of the genetic code and how inheritance works, which was the main subject of his research at the time. Of course, the genetic code is a long sequence of nucleotide bases, what is called DNA. And DNA is packaged into discrete units called genes, and it's the gene products that confer the function of living cells. Of course, genes are also packaged then into chromosomes that you inherit off your parents. So in the 100 years since Bateson, we're now in a position, after much research, and development to sequence DNA at massive scale. And that has given rise to the birth of an information science. You can imagine thousands, millions, or billions of these bases in long strings creates huge amounts of data. And actually, the generation of that data isn't the rate limiting step anymore. It's actually the analysis and the interpretation. But what can we gain from this? What can we learn from this information that we acquire through DNA sequencing?
With genomics, we can have the whole genome, so we can have all the content of this bacterium. We know which metabolite they are producing, to which target they are specific, and we can understand how they are acting to fight these harmful insects or fungi. We can find even new metabolites that are not investigated before with industrial importance.
Even if we find interesting results in lab or in field or under a greenhouse, but we cannot explain the how. It's an important part to understand the mechanisms. The work will not be complete. And we cannot advance the research to understand more without the genomic analysis.
For me, as a scientist in the field of biocontrol environment, knowing the interaction or the system of interaction between my bacteria and the plants is crucial. And we are trying to produce some biofertilizer and biofungicides with these bacteria to enhance and promote the production of crops. So knowing this interaction and genomic feature that enable my bacteria to interact with other plants is in the genome. So genome is the key.
This is the same philosophy that drove what is really now known to be a revolution in genomics. This was the Human Genome Project. So this was a 14-year, £4 billion endeavor that culminated in the publishing of the first draft whole human genome sequence. And that pursuit led to the development of state-of-the-art technologies that really have led to the democratization of genomics. And it also led to this dramatic reduction in the cost of DNA sequencing over the subsequent 20 or so years. So this truly was a genomics revolution. However, a revolution, it's only a revolution if it's equitable, if access to these technologies and the necessary training is available to all. And as we have seen, large swathes of the globe, of the research community, particularly on the African continent and in the Middle East, parts of South Asia and Central America, still do not fully harness the power of genomics in their research. And this was most recently felt during the COVID-19 pandemic. And this was data taken in the early part of 2021 that showed that the genomic surveillance of virus strains was particularly low in many regions around the world.
First challenge we have to face is the money. And I would say that it is particularly difficult for molecular biology and microbiology because we really need a lot of consumables. But it's not only for materials, it's also for publication. Now you need to pay for publication. So we really have a problem with this.
We faced a low budget dedicated for costly experiments, like the identification of the benefic bacteria and especially the complete genome sequencing. And also lack in technical expertise to analyze it.
It's too expensive to do only one whole genome sequence. It's around $1,000. That means more than 3,000 Tunisian dinars.
It's quite challenging for us when you are in a new field, for example, for the whole sequence, is to try how to analyze this data And sometimes the software are not free. Sometimes we don't really have the knowledge to do so. So this is quite the challenging that we face here in Tunisia in general.
Access to genomic new technology is really important for anyone, and for me especially, because it gave me the chance to launch myself in the genomic world.
We believe that part of the solution to this inequitable access is GetGenome, Genomics for All, the democratization of genomics. And so GetGenome was developed to empower, first and foremost, to help researchers overcome the barriers to access those technologies and fully utilize them in their research. Genomics is integral to life science research here in the West, so it should be globally. So GetGenome was conceived in 2019, and very quickly we generated some pilot studies. We launched those in Tunisia. They were highly successful, and they gave us the confidence to form a broader campaign, which was the GG Tunisia 2022 call for projects. In order to help conceive this campaign and to manage, we put together a very experienced team, a board of trustees, Apollinaire, Saskia, and Nick, who would join Sofian to help drive GetGenome forward. Shortly after, we registered with the UK Charity Commission, and that gave us then the confidence to move further afield and start our activities more broadly. So we launched a campaign in 2023 in Mexico and a similar campaign in Pakistan in 2023. But at the same time, we were engaged with the cohort, with the participants from the Tunisia call. We held a symposium, and we've been engaged with in a number of different conferences and seminars. 2024 has been very busy for us, so that in the first half of 2024, we launched GG Lat 2024. That is a trans Latin America call, and we have returned happily to North Africa and the Middle East, where I have no doubt we will be supporting many more Tunisian researchers as a part of our MENA call. A little look at the next step for GetGenome on this front will be a call in South Asia, which will happen likely late summer, early autumn. So in terms of our Tunisia call, I have to say that we were truly overwhelmed by the support that we received by the embassy, His Excellency, and all of the Tunisian research community, who really opened their arms and embraced GetGenome and have really made GetGenome what it is today. I have to say, without that community, we wouldn't have had the success and the confidence to move into other areas. So how does it work? Well, it's quite a simple pipeline, and it's bottom up. It should be remembered we help the researcher first. What we do is we conceive a campaign such as GG Tunisia 2022. We open a call for projects. We ask researchers around the country to apply for our support. Once they've obtained our support, then we ask them to deposit their organisms with us, and then we acquire the genome sequence, and we provide it back to the participant. The currency of research is publications. All research should be disseminated to the broader community and to the public in order to help other communities with their research and help the public more generally. So open science is an incredibly important aspect of that. So we encourage our participants to share their data, the data that we've helped them generate, onto open access, free to deposit, free to access platforms where they can gain credit for the work that they've done. Finally, network. As you can imagine, we've engaged with a number of communities, and as a part of GetGenome, we've started to bring them together. So we have a thriving alumni network, and we've found many opportunities to bring those researchers together. And what we often find is that they face similar barriers and they are interested in similar research themes and organisms, which is fantastic. So as a part of the Tunisia call, we supported directly 31 scientists. across 8 different institutes, but the number of scientists that we've actually interacted with is several-fold that. As a part of that work, we've generated the whole genome sequences of over 130 organisms, and we've engaged that community in 12 hours of training workshops, as well as conferences and symposia, etc. So some of those engagements include training workshops, many of which are online, and our symposium and various conferences. There are several funding streams for GetGenome. We're able to be attractive for small grants, and we're also interested in philanthropic donation. These are people that recognize that supporting early career researchers, helping to develop a thriving research community can only benefit the broader community. So I mentioned open science, and for those of you that are outside the realm of science, you might think that publicly funded research is accessible to the research community. However, that isn't often the case, unfortunately. We find that a lot of publication, a lot of data hides behind paywalls and subscriptions, and often that means that researchers who are in institutes that don't have the means to provide those subscriptions don't benefit, cannot access those publications. So open data, open access is our philosophy. That means free to publish their data and free to access data. And our participants have benefited from the availability of data, just as all of us in the research community have benefited from that. When asked, around 70% of applicants of our MENA call stated that these opportunities to publish data was their primary motivator, but also opportunities to network and collaborate, the access to the expertise and the technology. And of course, they want to improve their career prospects, overcome the limited resource, improve their data quality and impact, and also provide that information, disseminate that information that they've learned to their colleagues. And I agree with all of these. So I'm really proud of the Tunisian community, the research community that we supported through the Tunisia 2022 call, because they embraced open science fantastically well. We asked that they deposit their raw data on NCBI, which is a repository for genomic sequences, and they overwhelmingly did that. We asked that they write genome announcement publications that could then be cited by other researchers, and they did that as well. In fact, nearly 100 NCBI deposits have been made from this cohort alone, and 20 genome announcement publications have been published on a free to publish, free to access platform. And these publications are being accessed by the community, which is fantastic to see. So GetGenome supports a wide breadth of research. Some of the notable ones, bioremediation, bioengineering, probiotics. So research that is concerned with these themes. But overwhelmingly, in the region, in Tunisia, a lot of the research community is interested in plant growth promoting bacteria.
As you know, there is big challenges in agriculture now, especially with limited available resources, pollution problems, soil degradation, so the problem with pest management. So to face all of this challenge, there is a need of sustainable solution. I'm working on the use of biofertilizers and biocontrol agents based on bacteria, which can be part of an alternative and sustainable solution.
Even the climate change can affect the presence of the one insect or another, even the conditions of the product or the formulation of the products can be more suitable for an environment than another. So to have the specific product to the specific targets, it's very important. Many countries, they want to go for less use of pesticide. And on the other hand, they are encouraging the use of bioproducts. I'm interested in the microbial biopesticides, especially those produced by a bacterium called BT. Bacillus thuringiensis is a safe bacterium, and the products based on this bacteria are used for biological control.
So we are trying to build a consortium of microorganisms that will be helpful not only to promote plant growth, but to alleviate plant abiotic stress by using those microorganisms. When we irrigate with wastewaters, we are trying to reuse wastewaters
Agriculture, particularly in regions like Tunisia, is under immense pressure. And those challenges come about because of soil degradation. So desertification is a particular problem. The changing climate and the potentially harmful pesticides that farmers have relied upon to this point for controlling pathogens in the field. They're now being taken off the market, and in many cases, rightly so. And solutions now to tackle these challenges have to come to the fore. GetGenome is playing a role in helping supporting these researchers to find these solutions, and we're very proud to do so. So the solutions include these plant growth promoting bacteria. Our participants send us these strains, and we can generate the genome sequences and provide that data back to the researcher. We can help them try to analyze and interpret that data to develop the next generation of bioproducts and biocontrol agents and biofertilizers, etc. So I think I'll finish essentially where I started, in that promoting, celebrating collaboration between the UK and Tunisia, within Tunisia and within the UK, is always positive. GetGenome was developed as a vehicle to empower and to promote these partnerships and collaborations. And science research is an incredibly collaborative venture. Nobody stands in isolation. And so it's incredibly important that we continue to bring the community together to find sustainable solutions to these challenges that not just agriculture faces, but the public faces more generally.
Actually, when we receive our data, the whole genome sequence, at first we were, what am I going to do with it? I have a lot of data, I don't know how to start. So with my colleagues, my team, we did start to try to find how to analyze this data and what we can do with it. So it was quite challenging, but we persevered until we now have some knowledge. We know that a lot of students from CBBC and from Tunisia did also benefit from GetGenome. And maybe we can facilitate for them how to analyze their data. And so we conducted several workshops, and we did have a lot of participants from all Tunisia and from Algeria as well. And it was very empowering. So for my point of view, my experience, collaboration are really, really fantastic. And it's the way to really empower research.
I think it's always beneficial doing work together with other colleagues because we always learning more skills to analyze data, to discuss results. And if we stay in our laboratory and we don't make interaction with other people, we can't catch the technology.
We need to improve our skills and to be more independent in genomic analysis and sequencing, because it's the future. I think it's important when we do collaboration to be equal partners. So we can share equally, to be taken seriously with our partners, not because we lack some skills or expertise that we are not good enough. One of the best things with GetGenome, we are more independent in our genomic studies, and it's give us more resilience as researchers.